much, Alana. Thank you so much for coming today, guys, and then the Thrill Conference. It's been fantastic. I know Guy, Jenny, and I really loved seeing everyone here, and it's nice to kind of wrap it all off before we all head off to our wonderful worlds and to do more lab science. It's going to be great. So my whole project is about the P2Y6 receptor, and I'm going to be discussing it in the context of aging, and then I'm going to move slightly on to development, and hopefully you'll see the logical progression in why I've chosen to do this way. So to start off, in Guy's lab, in general, we love microglia, but in particular, we're really interested in this process or this function of microglia we call phagocytosis. Right? This eating of different targets, whether it be a whole neuron or let's say the synapse, like a subcellular target, or like myelin, and all these sort of different targets they eat are very important for ensuring you've got a healthy brain in a variety of different ways. And it's great the research that's coming up because of this. So in Guy's lab, we've been, for a long time, Guy's been characterizing how these microglia actually determine, how they decide whether to phagocytose something or whether not to phagocytose something. And it's done this via a variety of phagocytic signals. For example, you have an eat-me signal on the surface of an apoptotic cell like phosphatidylserine, which will then maybe bind to an opsinin, which bridges the gap between the eat-me signal and then the phagocytic receptor, which is on the surface of the microglia. And that's just kind of in general. And once the microglia has received that signal, it encourages the microglia to go phagocytose that target. But I'm not interested in phosphatidylserine. I'm interested in the P2Y6 receptor, which is just here, where you have the general idea that we believe in. What happens is when a cell is stressed, it releases UTP, which is then hydrolyzed to UDP, which activates the P2Y6 receptor on the surface of microglia. What actually is the P2Y6 receptor? Well, it's just a G-protein coupled receptor with its main ligands being UTP, but it also has affinity to UTP, ADP, and ATP as well, and with much lower affinity. And it's a pretty typical GQ, G-protein coupled receptor in terms of once the ligand binding induces PLC, induces a calcium rise, pretty typically there. But what's really interesting about this receptor, in the brain at least, it's almost exclusively found on microglia, which is really cool, especially if we want to look at phagocytosis in general. But why do we think it's involved in phagocytosis? This guy did a really good job yesterday, 2 days ago, analyzing or showing you some of the preliminary data. And to beat a dead horse, I will show you some more. So to start off, Kazumi and Inui back in 2007 showed that if you added UDP, that ligand for P2Y6, to primary rat microglia, you see a 10-fold increase in the phagocytosis of zymosan particles, showing that P2Y6 activation seems to induce phagocytosis. And not only that, if you add canic acid, which is an excitatory stress, to neurons in vitro, you see the release of UTP, being the sort of pre-hydrolyzed ligand for P2Y6. And not only that, if you do it in vivo as well, you can actually inject an IP, which is bizarre. You can also see an increase in the amount of UTP production in the brain. This is sort of like a dialysis system taking the seeing the solution as it was coming. And you see quite a large increase in UTP as well, showing that when neurons are stressed, at least in excitatory stress, you see the release of the ligand for P2Y6, at least the pre-hydrolyzed ligand. Guy's lab then wanted to see this further. He wanted to explore this as a potential, or see, could we look at this therapeutically, or is there a potential therapeutic angle for this? So using the CGT cultures, which Tim talked about a lot in his talk just in the last session, where you have a triculture system with neurons, microglia, and astrocytes. They're really fantastic culture, especially at looking at sort of this neuroinflammation-induced model, because if you add LPS to them, you often will see very clear and microglial-dependent neuronal loss. But in this situation here, the addition of 100 micromolars of UTP actually induces a significant loss in neurons because of that. And actually, if you add MRS 2578, which is a specific inhibitor for P2Y6, you don't see that loss. You see protection from that loss, showing that the UDP-induced loss is P2Y6 dependent. And not only that, if you add LME, which is a nice way to deplete microglia, you also don't see the UDP-induced loss, showing it's a microglial-dependent loss as well. This is also then followed up later by an ex-postdoc in our lab, looking at wild-type and P2Y6 knockout CGC cultures as well, showing a very similar thing, where if you add UDP to wild-type cultures with microglia with the P2Y6 receptor, you do see neuronal loss, but then you don't in the knockout cultures. This is then followed up again. I love this, don't we? In more CGC cultures, this, like I said, this is a really great model for neuroinflammation. If you're adding LPS, you see a microglial-dependent neuronal loss. And actually, if you co-treat LPS and MRS, that inhibitor for P2Y6, you see a quite significant reduction in neuronal loss in these CGC cultures. So that's both for blocking P2Y6 and, again, if you use P2Y6 knockout mice as well, showing that actually P2Y6 seems to be involved in a neuroinflammatory-induced neuronal loss. This is then followed up further by looking instead, this is in actually a tau model of mice using P3ONS mice, which we collaborated with Maria Spilantini. And actually, if you cross P2Y6 knockout mice with P3ONS mice, which have a really kind of gross tauopathy, and don't even survive past 7 months really because of the spine, the issues in the spinal cord. But Ma was able to see that actually, if you look at, first of all, if you look at the behavior, just here on the right, you see a substantial amount of cognitive decline. This is a novel object recognition test. And you see a reduction in memory just in the wild-type mice with these P3ONS mice. But if you knock out P2Y6, you see complete protection from this memory loss. And not only that, though, you see a slight protection in neuronal loss. But I really want to draw your attention to the fact that actually the neuronal loss is really tiny. It's a really small amount of neuronal loss. This is in the parahindal cortex, a region which is really associated with this sort of the novel object recognition test and is quite associated quite well. But a very small amount of neuronal loss is needed for quite a substantial loss of memory, which is really quite interesting. And got Guy thinking about whether, okay, if neuronal loss doesn't seem to be relatively small in comparison, is there another target that's being phagocytosed or removed first prior to neurons? And finally, just to kind of go through this is a lot of people have seen this graph or a version of this graph at some point, showing that amyloid plaques seem to be very much before occurring way before in Alzheimer's disease. And actually, what correlates much closer with memory loss or the symptomatic situations of Alzheimer's disease is actually this sort of decrease in brain structure or this dysfunction in brain structure, whether it be neuronal loss or potentially synapse loss. There's actually those connections between neurons. So that in mind, this is kind of this is where I kind of started my PhD. This is where I was going, and the general idea that UDP the neuronal stress induces the release of UTP, activating P2Y6, which would cause the phagocytosis of some particular target, which we didn't really know about at the beginning. And so I was to investigate why this is kind of occurring. And actually, what I wanted to particularly look at is in the context of aging. About 40% of people over the age of 65 will experience some form of memory loss in aging. So I wanted to investigate that further. Is P2Y6 a potential therapeutic angle there? If it's already shown protection in a neurodegenerative model, we make it show protection in an aging model as well. So to do this, I was looking at a couple of different sort of models. First, we're using the CGC cultures. This is mainly Tom, an ex-PhD student in our lab, but also isolating primary microglia from the brains of wild-type and P2Y6 knockout mice. And then also doing some cryostats, doing some immunohistochemistry on some free protein brain sections, and see if we can sort of tease out what might be the mechanism for any sort of effects you see there. So to start off, I wanted to check to see if phagocytosis in general was affected in these P2Y6 knockout mice. We had some evidence before that it wasn't, but I just wanted to try with, this is with phosphatidylserine-coated beads, where if you add these to microglia, you wait about 2 hours. You can then use flow cytometry to determine how many of these beads have been uptaken by the microglia. And if you do this, you see no real difference between the wild-type and the knockout mice, suggesting that phagocytosis as a function isn't being dysregulated by the fact there's no P2Y6. However, then if you add synaptosomes, being isolated synapses from mice or rat brains, and you can label them with pH RODA, a pH-sensitive dye that only will fluoresce red once it's in the lysosome, the acidic lysosome, you actually see a significant reduction in phagocytosis in P2Y6 knockout mice, suggesting that P2Y6 is important for the phagocytosis of synapses. Not only that, so this is with Tom's work from before, culturing those CGC cultures that Tim showed us lots of lovely pictures of, you can actually add a slightly lower dose of LPS to what he was adding before, where you don't see neuronal loss, but actually you can see a reduction in synaptophysin staining, so a reduction in the amount of synapses connections between these neurons. However, you do see this loss in wild type, but not in the P2Y6 knockout mice, which is very interesting. I will obviously note as well, the levels do seem to be, they weren't significant, but they do seem to be lower in the P2Y6 knockout mice, which is kind of an interesting thing to note. I might point out later. So then, obviously, all these in vitro models are fantastic, and they're very, very helpful, but actually we should look inside the brains of these mice as well, and see the actual system and how it works, and see if we can find a mechanism there. So to do that, I was looking for the co-localization of pre- and post-synaptic markers, being vGlut1 a presynaptic marker and HOMA1 a postsynaptic marker, and looking for any changes in synaptic density in a variety of different regions, especially associated with memory. So to start off, I still looked in the somatosensory cortex and the CA1 and the hippocampus, and I saw a decrease in synaptic density, in an aging-dependent decrease in synaptic density in the wild-type mice, but no such decrease in the knockout mice, which is really interesting, showing that that P2Y6 seems to be important for this age-induced synaptic loss. I will obviously point out, though, in the somatosensory cortex, it does seem to be slightly reduced synaptic density in the young P2Y6 knockout mice. This is kind of interesting. However, just because there's synaptic loss, we need to try and link it more clearly to microglia, is microglia particularly involved with this? So to do this, I wanted to look at microglial phagocytosis in the three associating bone sections. So to do so, I simply labeled with IBA1, a microglial marker, CD68, a lysosomal marker, and then vGlut1, the presynaptic marker I mentioned before, and looked for the internalization of vGlut1 within CD68, the lysosomes of microglia. And then doing something similar to what Ryan from Cardiff was doing earlier, doing some sort of engulfment analysis. We're using IMRES to generate surface-rendered images. And if you do that, you see an age-dependent increase inside CD68 volume, which is great, because I think we've literally just shown that in the, in the blood vessel talk just a second ago. And that's expected. That's relatively well characterized, and you do see an increase in CD68 volume in aging mice, showing that phagocytosis, there's still an age-dependent increase in phagocytosis, and that seems to be independent of P2Y6. However, if you look at vGlut1 internalization within CD68, within the CD68 of microglia, you see a significant reduction. And you compare the old mice of wild-type mice and P2Y6 knockout mice, showing that P2Y6 seems to be important for the age-induced increase of synaptic material. And to validate all this, and to look into this a little bit further, Ma also showed that in the novel object recognition test, you see a clear reduction in memory, which has been very much reported before in wild-type mice, but in P2Y6 knockout mice, you don't see that reduction at all. However, it's very small, but there does seem to be a slight reduction in memory, or at least worse off in this novel object test in the young mice. So just to kind of conclude that first section, it seems that P2Y6 does seem to influence the phagocytosis of synapses, especially in age-dependent manner, and through the phagocytosis of synapses, or microglial phagocytosis specifically. And secondly, or thirdly, I guess here, P2Y6 is involved in the age-associated memory loss as well because of this. Thank you. But that got me thinking, okay, well, is microglia, we see some of these effects where synaptic density seems to be slightly lower in the younger mice compared to the older mice. So that got me thinking, is there potentially a role for P2Y6 in development as well? So to just kind of give you guys a general idea, synaptic pruning during development, you start off with X number of neurons, and then that, synapses, and that explodes all the way throughout sort of development, and then it'll start to wean off as you get older. And the reason you have so many of these, and you have a lot of synapses, and your body really can't maintain this amount of synapses, and they're removed in an activity-dependent manner, partially through microglial phagocytosis. So I love this microglia. So I wanted to investigate, is P2Y6 involved in this process in any shape or form? And to do this, I looked at P30 mice. This is a key sort of developmental time point in mice where synaptic pruning is still readily occurring in these in the mice brains. And again, looking at synaptic, just doing similar to what I did before. First, we do see doing the surface rendering and looking at the internalization of vGlut1. First of all, I do see a slight increase in IBA1 volume of these microglia at P30. I checked any sort of morphological changes as well. I didn't do Sandra's technique, I wish I did. But I didn't see any sort of changes in endpoints, branch points, or the mean, maximum, minimum sort of length. So it didn't seem like there's many other morphological changes occurring there. But actually, if you look at the CD68 and the vGlut1 internalization within CD68, you do see a significant reduction in synaptic material in the P2Y6 knockout mice. showing that P2Y6 is essentially involved in synaptic pruning, and without that signal being activated, you do see a reduction in the phagocytosis. Then I wanted to check synaptic density in a variety of different regions associated with memory at this sort of age. And in 3 out of these 4 regions I've looked at, you do see a significant significant increase in synaptic density, which kind of correlates to the reduction in synaptic phagocytosis that we're seeing. And that kind of makes sense. I will note that obviously the change in density is relatively small, but in the necessary respect, a big change, a big or small change at this time point could be very, very important and could have an exponential change later on in life. So we wanted to follow this up slightly further and look at adult mice. This is the work of Stefan and Mar, who used to be members of our lab, testing memory adult mice. And you see, we're just doing this is in long-term memory, sorry. So doing the novel object recognition test, as well as doing the object location memory test. And in both situations, P2Y6 knockout mice performed slightly worse and significantly worse than wild-type mice. Then also looking at short-term memory tests using a Y maze, as well as a novel object recognition test. This is a much shorter time period of just 2 minutes. You also see a significant reduction in memory. Now, I will point out, obviously, there's still evidence, clearly, that these mice do remember and they are able to perform the tasks well, to some extent, but they're definitely not performing them as well as the wild-type mice, suggesting there is some sort of dysfunction there happening in memory. So I need to conclude, so I can let you guys all leave. P2Y6 is involved in seems to be involved in synaptic pruning during development, and actually this P2Y6-dependent synaptic pruning during development might be important for maintaining synaptic density in a variety of different regions associated with memory. And finally, this synaptic pruning may be important for memory during adulthood, although it's very hard to tease out the differences between, does the microglial P2Y6 receptor have an active role in memory? And that's quite hard to do without sort of some live imaging techniques and whatnot. Thank you so much for listening. Thank you so much to the Brown Lab, and I'll take any questions.
Brilliant. Can we please have one burning question? Anybody? Jenny.
Oh, am I on? Yeah, I'm on anyway, in a minute. Can you just speculate, so in development, if you don't have P2Y6 receptor active, mediated synapse stripping, what might be the sort of developmental problems that you might get?
So obviously there's some evidence pointing towards dysregulated microglial phagocytosis in sort of like some neurodevelopmental disorders such as autism or schizophrenia, where you have excessive or insufficient amounts of pruning. It's difficult to understand, right? Because I wonder these processes need to be very tightly regulated, and all these different signals we've evolved, and all these different signals are very important to involved. But obviously these microglia, these mice never had P2Y6 as well, and there's also a lot of evidence that a lot of genes are redundant as well. Something I've always kind of thought about is whether these receptors, especially on microglia, sort of help them decide how to spend their time. They have a signal that tells them you need to go and eat synapses. They're not real. Microglia don't have feelings, but it's the easiest way to talk about them. It tells the microglia to go eat a synapse. So because it doesn't have that signal, if it's spending less time eating a synapse, is it spending more time eating something else? So while synapses are being affected in some way, and that may be detrimental, it could be detrimental here. They may eat more myelin, for example, or they may eat something else instead, and that might be also causing an issue. So I've also tried to look into that a little bit here, but I kind of ran out of time to talk about any more. So,
thank you, Jake. Brilliant. Let's please thank.
Don't go.
Don't go, but let's have a final big thank you to all the speakers. Thank you.